1. Introduction to motor neurons. Definition. Motor neurons, or efferent neurons, are nerve cells forming part of a pathway along which impulses pass from the brain or spinal cord to a muscle or gland. Directionality. Unlike sensory neurons, these carry signals away from the central nervous system (CNS) to the periphery to initiate movement or secretion. 2. The structure of an efferent neuron. Multipolar shape. Most motor neurons have a multipolar structure characterized by a single long axon and many dendrites that allow for extensive integration of signals. Soma location. The cell body or soma is typically located within the gray matter of the spinal cord or the motor cortex of the brain. Axon length. The axon can be incredibly long, sometimes extending over a meter to reach the distal muscles of the feet. 3. Upper versus lower motor neurons. Upper motor neurons. These originate in the cerebral cortex and travel down to the brainstem or spinal cord. They are responsible for planning and directing voluntary movement. Lower motor neurons, LMN. These carry the signal directly from the spinal cord to the effector muscles. They serve as the final common pathway for all motor output. Synaptic connection. The UMN synapses onto the LMN, creating a relay system that translates mental intent into physical action. 4. The neuromuscular junction, NMJ. Synaptic cleft. This is the microscopic gap between the motor neuron terminal and the muscle fiber where chemical communication occurs. Acetylcholine release. When an action potential reaches the terminal, it triggers the release of the neurotransmitter acetylcholine, ACH, into the cleft. Motor end plate. The muscle's specialized membrane contains receptors that bind to AC, initiating a muscle contraction sequence. 5. The motor unit. Definition. A motor unit consists of a single motor neuron and all the individual muscle fibers it innervates. Recruitment. The body increases the strength of a contraction by activating more motor units, a process known as recruitment. Fine versus gross control. Small motor units control precise movements like typing, while large units are used for powerful actions like jumping. 6. Somatic versus autonomic motor nerves. Somatic system. This branch controls voluntary movements of skeletal muscles, such as walking or waving. Autonomic system. This branch controls involuntary actions of smooth muscles, cardiac muscles and glands, regulating functions like heart rate and digestion. Effector differences. Somatic signals go directly to skeletal muscle, while autonomic signals often pass through a ganglion first. 7. Signal transmission. Action potentials. Electrochemical nature. The signal travels as a wave of depolarization along the axon membrane, driven by the movement of sodium and potassium ions. All or none law. A motor neuron will either fire completely or not at all. The strength of the stimulus is coded by the frequency of firing, not the size of the pulse. Saltatory conduction. In myelinated motor nerves, the signal leaps between nodes of Ranvier, significantly increasing transmission speed. 8. Myelination and speed. Schwann cells. In the peripheral nervous system, these cells wrap around motor axons to form the insulating myelin sheath. Conduction velocity. Myelination allows motor commands to travel at speeds up to 120 meters per second, ensuring near instantaneous reactions. Protection. Beyond speed, the myelin sheath provides structural support and protection for the long, delicate axonal fibers. 9. Common motor nerve pathologies. ALS, amyotrophic lateral sclerosis, is a progressive disease that causes the degeneration of both upper and lower motor neurons. Neuropathy. Damage to peripheral motor nerves can lead to muscle weakness, fasciculations, twitching, and eventual atrophy. Myasthenia gravis, an autoimmune disorder that disrupts the communication at the neuromuscular junction, causing rapid muscle fatigue. Regeneration and atrophy. Wallerian degeneration. When a peripheral nerve is cut, the part of the axon disconnected from the cell body breaks down and is cleared by Schwann cells. Schwann cell tubes. Surviving Schwann cells form a regeneration tube that guides the new axonal sprout back toward the target muscle. Denervation, atrophy. Without constant nerve stimulation, muscle fibers shrink and lose protein content, eventually being replaced by connective tissue, if not re-innervated. 11. Transmission speed, muscle versus gland. Direct myelinated path. Somatic nerves to muscles are heavily myelinated and direct, prioritizing high-speed 120 meters per second transmission for rapid physical response. The relay delay. Autonomic paths to glands use a two-neuron relay. The synaptic pause at the ganglion and lack of myelin on the second neuron result in much slower speeds. Biological efficiency. Slower speeds are sufficient for glands and smooth muscle, as these targets regulate steady-state physiological functions rather than emergency movements. 12. Conclusion and summary. Functional essentiality. Motor nerves are the bridge between the digital processing of the brain and the mechanical output of the body. Complex coordination. The integration of UMNs and LMNs allows for the incredible grace and power seen in human movement. Future research advances in neuroprosthetics and regenerative medicine aim to restore function to damaged motor pathways.